Now we're going to start with the nervous system, and this chapter is probably one of the more challenging ones that you're going to have just because of the amount of physiology that's in here. There's a lot of memorization, but we'll get through it. Alright, so what your nervous system is for is obviously to respond to your environment. So that's going to be the sensory input that comes in, and everything that our body reacts to, whether it's something that you think through or feel or physiologically do, is due to sensory input. So it's going to involve things that happen with what you see, what you hear, temperature, touch. That's all sensory things that are going to be coming in. And then where things are processed is going to be in your brain and in your spinal cord. And so that's called integration. And then what your body ends up doing, or the action that you take, is considered to be the motor output. So those things are going to be either voluntary or involuntary. The involuntary piece is the majority of what's taking place inside you in terms of the physiology of how your body's going to respond. And those are the things that our autonomic nervous system takes care of, which is our parasympathetic and sympathetic. The only voluntary things that you actually have are your skeletal muscles. You're the one who decides when those are going to move. But they're all based on, obviously, things that you are reacting to. So when you look at sensory inputs, for example, you see something, right? So you're going to have light waves that are going to be coming in through your eye. What you see is processed in your brain in the very specific areas that vision is integrated in. And then what you're going to do is going to respond based on what the action is. So that's why we call the sensory portion is the affector, and then as you process it, and then the motor output comes in, and then where it results is going to be the effector. So our nervous system is set up by having the central, which is your central processing system. It's your brain and spinal cord. And then you have the piece that's called the peripheral nervous system. The peripheral nervous system is outside of the brain and the spinal cord. You're going to have what we call sensory information coming in, and that is called afferent, or it comes in through afferent nerves, which are your sensory nerves. And then once it gets processed up here in the spinal cord of the brain, then you have the motor output, and the motor is the efferent. The motor is then divided into what we call the somatic motor, which is the voluntary portion that involves your skeletal muscle, and then the autonomic portion, which is the involuntary, and that is going to involve your cardiac, respiratory, your smooth muscle, your glands, digestive, urinary, reproductive, all the things that function automatically. And from that comes your parasympathetic and your sympathetic branches. So the parasympathetic, of course, is known as rest and digest, which are the homeostatic at-rest mechanisms that keep us healthy. And then the sympathetic is what kicks on when a stressor is sensed, whether it's a threat or it's something that you have decided that you're going to do. For example, when you're working out. So your body's going to start physiologically producing more carbon dioxide, more acidity. And so then your body has to respond to that. And the body says, hey, this is now a challenge to our physiology, so we're going to respond by activating sympathetic functions. So it isn't always that it's going to be some kind of thing that would be considered to be a threat. It just means that there's now some instability that has occurred in terms of what your body is feeling. And then the sympathetic system is going to kick on. Anytime your sympathetic system kicks on, it's going to inhibit parasympathetic responses. And that's very important that you remember that. Because any time that sympathetic kicks on, that means that your rest and digest, your resting homeostatic functions, are no longer going to be activated. We need to respond to a challenge, and that's why sympathetic always will dominate parasympathetic when it senses some kind of threat or challenge. And then parasympathetic is what will then return everything back to normal. So when we get to talking about the heart and what happens with respiratory rate and all that, we will then get into how that works. So structurally, our central nervous system is made up of our brain and our spinal cord. And again, that is going to be your integrating, your command center that is going to regulate everything. In our peripheral nervous system, we have the spinal nerves that come off the spinal cord, and then we have cranial nerves that come out of the brain. And so those pieces that exit from the brainstem, which is where the cranial nerves are passing through or originating, those are going to be the pieces that are going to regulate a number of different things that are then tied to our homeostatic functions. The whole key of our peripheral nervous system and what the nerves do is communicate. So you're always going to have some kind of sensory function that's taking place, and then you're going to have a motor function of what then responds to what it is that it received. So when you look at the spinal nerves, we have 31 pairs of spinal nerves. And when we get to talking more about the spinal cord, I'll get more into how it is that they function. But here I just wanted to give you the example of where our spinal cord is located, obviously within the vertebral cavity that is formed by the vertebrae when they stack up. So our spinal cord is really well protected. But coming off of that, peripherally, so that means they go out to the side, so they're going away, you're going to have 8 cervical, 12 thoracic, 5 lumbar, and then 5 sacral, and then 1 down in your tailbone that's called the coccygeal. And so these nerves are going to exit up above the vertebrae under which they're numbered. So we'll be getting more into these later. The cranial nerves are 12 pairs, so we have them, 12 on the left, 12 on the right, and they are numbered by using Roman numerals, and they each have obviously their own specific function. But the key thing here is that they actually come off the brainstem, which is this portion here of the brain, and we'll get into the details of how these work a little bit later. Then our nervous tissue, the way that it's set up, we have 2 types of cells. We have what are called neuroglia, and then we have neurons. The neuroglia is what I like to refer to as the support staff. So these actually support the neurons and protect them. And then the neurons themselves are the cells that are going to receive information and then send out information in terms of what it is that needs to happen. So we have neurons within our brain, and then we have neurons that are going to originate in our spinal cord and then go on out. So I'm going to start with the neuroglia. So these are your support cells. The first ones are called the astrocytes, and the astrocytes form the blood-brain barrier. So if I ask you a question about what is the blood-brain barrier, always remember these are the astrocytes. So I'd like to think of them as the brain bouncers. And what they do is that they surround the capillaries, which are the blood supply for the brain, and you'll see that they kind of like sit, the actual body of the cell sits kind of in the middle between capillaries, and then has basically hands or paws, is like I like to think of them, that wrap around the capillaries, and then also parts of them will wrap around pieces of the neuron. And so what they do is they actually protect things from coming through those capillaries. So only substances that can actually diffuse and fit through the little gaps between the paws are actually going to get into the brain. And then what they also do is they help reset the polarity of the neurons based on what is happening with, sodium and potassium function. The microglia, these are immune cells that actually live within our brain, and I like to kind of think of them as little roaches, cucarachas. And what they do is they help clean up any waste or any damage that occurs, and they're very, very important in maintaining the health of the brain, because immunity is absolutely critical of what takes place within our brain. And when we talk about the lymphatic system and our immune system, we'll again talk a little bit about these guys and what their role is. But for now, what I want you to remember is that the microglia are the immune cells that live within our brain. Then the ependymal cells, they look like little goats, and what their job is that they actually line the spaces of the ventricles and help produce the cerebral spinal fluid. So they're very important in doing that and secreting that CSF into the ventricle space within the brain. And then that CSF obviously is what helps provide the buoyancy and the fluid environment that the brain is in, and also provide the CSF that goes down into the spinal cord. So it's a very, very important job that these ependymal cells have in making the cerebral spinal fluid. Then the cells that are called oligodendrocytes and the cells that are called Schwann cells, these produce what's called myelin. So first with the oligodendrocyte, you'll see that it has a big cell body, and then it has multiple branches that reach out from that cell body, and then what they do is this myelin that is produced wraps around the actual axons of the cells that are within the brain. So our neurons, the axon portion, and we'll get to that in a sec, is protected by these areas that have myelin. And why myelin is so important is that it helps speed up the signal that travels along an axon. Areas that have myelin are always going to be faster in terms of the signaling mechanism, and areas that do not have myelin are going to be a little bit slower. So what you need to know from these is that the oligodendrocyte is what produces myelin inside the central nervous system. outside in the peripheral nervous system, so these are the nerves that are going to leave and go out from the spinal cord, these cells that produce the myelin in the peripheral nervous system are called Schwann cells. And so what they do is that they're actual individual cells rather than one cell that wraps around many axons like it does in the brain and spinal cord. These are actual individual little cells that wrap around the axon that ends up leaving or coming in to the spinal cord. And so what happens is that each one of these will form like a little sausage looking thing, and then what we have is this wrapping that occurs. So it kind of looks like a hot dog bun around the actual hot dog. So if you think of the axon piece as a hot dog, and then the myelin piece of the Schwann cell that wraps around, that is your bun. And so what it does is that in having each of these individual little Schwann cells all along the axon, there are little gaps that occur in between. And why we have these little gaps is because when it's myelinated like this, it actually allows for the signal to travel much faster in that myelinated piece. And so that's why these are so critical. Then these satellite cells, what they do is they actually surround and protect the actual body of the neuron, and so they're more of just a protective effect around that. So think of it as having like a little helmet. All right, so our neurons themselves. So what you're looking at here is what, for the most part, a neuron looks like. Right? And so what you're looking at are these things that are called dendrites. So that's what looks like the hair that is coming off the head here. So this is the body of the neuron, what is called the cell soma. And then these pieces that stick out of it, these are your dendrites. And so their job is to receive information. So you could have over here another neuron that is connected to here, and then it sends information to this piece. And it has receptors that receive the information. And the information that I'm talking about is always in a chemical form. And the chemicals are called neurotransmitters. So the signal comes in, receives the information, then it travels into this body, to the cell body, the soma, where the information is actually processed. And so it's processed based on what chemical landed on here, and then this soma is going to respond. Then what happens is that signal is going to continue and get to this part that is the beginning of the actual axon, which is called the axon hillock. So this is where the signal has to get to if you're going to generate what's called the action potential and then travel down the axon, which are these pieces that you see coming down. So this is an axon, so it's taking the information that got processed here in the soma, and then it leaves and it's going to travel down. Here's another branch of an axon. But once the signal travels down, it's going to reach what is called an axon terminal. And in this axon terminal, what we have are little containers that are called synaptic vesicles that contain the neurotransmitter. And so then that neurotransmitter will get secreted, and then this axon here that is leaving is sending the signal to something else. So it could be another muscle, it could be a gland, it could be, if this was within the actual brain, obviously you wouldn't have Schwann cells in these, but the axons that are within the brain are then sending signals to other neurons. So what you see down here, this is what an actual neuron with the cell body, and then you've got the little satellite cells around them, and then this is the axon that's leaving, and then this is your neuron. And then all of these things coming into the top of the soma, those are going to be the dendrites. So this is just the general structure of what you see within an axon and a neuron. So neurons again, nerve cells, and the dendrites are the sensory piece of the neuron. The soma is the cell body. The little dots and stuff that you see that are all these little dots here, these are called Nissl bodies, and what they are just rough ER, and you know that from the prior chapters where we talked about what does endoplasmic reticulum contain. It has ribosomes that make proteins. The axon carries the motor signal away from the soma, and then we have to get to this area over the axon hillock, which is where the trigger zone is, and that is where you're going to generate your action potential. So going back to what happens with the neuron, signal comes in, soma processes the information, it gets to this area where the axon hillock is, which is also where the trigger zone is, and just like a gun, when you pull that trigger, that bullet fires, boom, it's going to get fired and it's going to go down through the axon and then travel to the end here where the axon terminal is. And then from here is where you'll now have neurotransmitters that get secreted. Then what you also see on here where we have the Schwann cells, in between I talked about these little gaps. And these little gaps that are in between each of the Schwann cells, they are called nodes of Ranvier. And Ranvier, I'm sure, was the dude that discovered it. And what we have here is that there is a very high concentration of sodium and potassium gates. So what you learned in the prior chapters where we talk about depolarization and action potentials, that is what is happening here. So at the end of the axon, we have the axon terminals. And so this right here is a zoomed in view of what you have within an axon terminal. So inside we have vesicles, and in the vesicles is where the neurotransmitter is located. So it could be norepinephrine, it could be acetylcholine, it could be, GABA-aminobutyric acid. There's a number of different neurotransmitters that are made within axons, and they all depend on what the job of that particular neuron is. The axon terminal is the same thing as that synaptic knob, so this is your little knob, and then what you're seeing here is the junction between the axon terminal and then whatever that tissue is that it's connected to. And then there's a little space in between, and then these little purple things that you see are called receptors. So you've got your neurotransmitter, gets across the space, which is the synaptic cleft, right? A cleft is just the gap between the junction, and the junction is called the synapse. And then you've got your little neurotransmitters. They go across, and then they attach to the receptor. So this is the cleft, the chemical crosses, and then we'll make a connection. So remember that a junction, think of it as just an intersection where two things come together. So here comes your signal, release the signal, which is going to be the chemical, the neurotransmitter, that's going to cross, and now it's going to get across and find a receptor. And what's really important about receptors is either they are going to cause stimulation on the membrane that they are located on, or the chemical can actually inhibit the stimulation and inhibit the function of the actual cell. And what that does is that it prevents these guys from actually depolarizing. So we're going to get into that in a sec. Then what I want you guys to kind of think of as well is that when we have our central nervous system, we have little clusters of cell bodies, so little clusters of heads of neurons, all right, that are called nuclei. All right, they're very specialized areas. So I kind of think of them as little centers of really important thinking that takes place. Then we also have these things that are called ganglia, all right? And ganglia are actually little swellings in a nerve. Tracts and nerves are basically the same thing. When we have bundles of axons, we call those tracts in the central nervous system, so in the brain or the spinal cord. In the peripheral nervous system, we refer to them as nerves, all right? And then white matter is where you have a lot of myelinated axons, so that's why it looks whiter. And then gray matter is where you have unmyelinated fibers and primarily the cell bodies. Then again, to remember that the sensory portion is the afferent. So afferent always means it's going to the center. So think of it as when you're on the ambo or on the fire truck and you're returning to the station, right? The station is your center. So that is bringing in the information. And then the motor is the efferent, or what is going to go away. So think of the motor after you've received a call, and now you are leaving through the efferent track to go to the response of what you're going to be going to. Interneurons, these are neurons that are kind of in between, right? So what they do is they actually sometimes manage the signal of what's happening. So as an example, this is what your spinal cord looks like. So we have white matter and gray matter in the spinal cord. And what happens is you'll have information coming in from a sensory neuron. So this is what a sensory neuron looks like coming in from the periphery. So say this here was connected to your finger and you touch something hot, right? So you don't want your finger to burn. So what's going to happen is that information is going to roll in through the sensory neuron, come in through what's called the dorsal root, and I'll get into that later with the spinal cord, and it's going to bring in the information to the spinal cord, and we have a little interneuron that basically says, yep, this is important enough for us to very quickly send the signal back out through this efferent neuron, the motor neuron, boom, and then this is going to go to the muscles to make you pull away your finger and have basically a reflexive mechanism. So these interneurons are just neurons in between the sensory and the motor neuron, right? Sometimes you'll find these interneurons that will go up into the brain, and I'll show you guys examples of that in a little bit, but all they're called is just in between neurons. That's why they're called interneurons. So your neurons obviously are going to receive information. So at the end of all our skin areas, right? Within the skin, obviously we have a layer called the epidermis, which is the outermost, and then just below that is the dermis and then the hypodermis. And within the dermis, we have these little nerve endings, all right, that are actually going to feel stuff that is happening on our skin. And so if you cut yourself, right, when you're cooking, bad day at the station, obviously that's going to hurt, right? We've all done this. So what do you do when this happens? You pull away the thread, right? You take the knife out of the way and you feel that. And so that feeling is a free nerve ending that you're actually receiving that information. We also have these other things that are called Meissner's corpuscles. They're just touch receptors. They tell you whether something is soft, whether it's hard, whether it's rough, whether it's smooth. We have pressure receptors as well that are going to tell you how much pressure is being applied, and in some cases it can actually cause pain. And then within our tendons and our muscles, we have very specific receptors. In the tendon, we have what's called a Golgi tendon organ, and it also helps you measure the amount of tension that is within a tendon to then relay that information in to the spinal cord, to the brain, to then send the signal out, to then manage what's taking place within that. So sometimes you have to relieve some of that tension, or you may have to activate other muscles in terms of what's taking place to help stabilize a particular joint. Within the muscle tissue itself, in our skeletal muscle, we have these other pieces that are called muscle spindles, and they're also proprioceptors. And proprioceptors basically measure elasticity, tension, contraction. What is the pressure, the lengthening or the concentric, the concentration or the shortening of the pressure and force that is being generated in these tissues? And these are protective mechanisms so that you don't injure them. Then in terms of what the neurons look like, the one that I just showed you was called a multipolar neuron, but we also have what are called bipolar and unipolar neurons. So what they look like, this is what our multipolar neuron is going to have multiple poles. So that means you have dendrites, you have your soma, and you have an axon. So this is the majority. These multipolar neurons are what we have the most of. The bipolar neuron, this is what I just showed you earlier, and here when it's coming in and receiving the information, this is a perfect example of what's called a unipolar neuron, where the info comes in and then it has this one pole that just continues, right? And this is a big giant axon that's bringing in the information and then gets processed here in the little soma and then receives it here in the little interneuron. And then the bipolar neuron, these are similar to what you're going to, this is the bipolar here, the unipolar is down here. The bipolar neuron, what it's going to do is it's going to receive the information that is coming in, for example, in your eyes. We have these in our retinas. And what they do is they transfer the info that is going to then get sent to your optic nerve so you can actually see. And then here's that unipolar neuron that I talked about that will be bringing in like sensory information from your skin and then bringing it to the spinal cord so that you can actually process what you actually felt. All right, so the functional properties. So this piece of irritability is extremely important because this is how our neurons are able to respond to a stimulus. And what happens is that it converts that to a nerve signal. And the nerve signal actually generates an electrical current. So when you guys think of what is happening between the ions, the neurotransmitters, and what happens on the membrane, whenever you see the word signal or threshold or action potential, what is happening is actual electrical activity that is going to occur. And then conductivity is that electrical activity of the signal that is passing through either between neurons, from a neuron to a muscle, or from neurons to glands. So the first thing you need to understand is that we have what's called a resting membrane potential. So what that means is that a cell is unstimulated, but what we have is a plasma membrane that is polarized. And what that means is that there's an electrical difference. Just like with a battery, you have a negative end and a positive end. We have the exact same thing on a membrane. So the polarization occurs between positive ions on the outside, and then inside we also have positive ions, but there's less. And so what happens is that the inside of the membrane, due to the fact that it has less positive ions, you have negativity, right? So it just has lesser of a charge. So resting membrane potentials kind of look like this. We have sodium on the outside, so we have the majority of the sodium is going to be on the outside. And inside we have potassium. So even though potassium is a positive and sodium is a positive, what happens is the proportion of positivity on the outside causes the outside of the membrane to be more positive, which is why this is a plus, and the inside of the membrane, because it has less positive ions in terms of its proportion, it causes the inside to be more negative. What we also have inside the membrane is going to be anions, so it could be chloride, it could be phosphate, right? It could be bicarbonate. Those things are negatively charged anions, and so I just gave them an AN. AN doesn't exist, it's not an element, but it's just referring to an anion. So what you end up with is positive on the outside, negative on the inside. So now when you want the polarity to change, which means the outside will now become negative and the inside will become positive, we have to do something to the cell membrane. We have to stimulate it. So what's going to happen is we have this thing called depolarization. Depolarization means you're going to get an inward rush of sodium ions. And when that happens, you are now depolarizing and changing the inside of the membrane to more positive. So what occurs when you look at depolarization is you have a stimulus, right? So you'll have maybe this receptor here would have a neurotransmitter that causes stimulation. So think of it as maybe your heart. Okay, so your heart has norepinephrine, right? Now we got to speed it up. So you land norepinephrine on here, that causes this little gate to open, and sodium is going to start coming in. The region in here where you see sodium starting to come in over here, what is happening is that it's starting to change the inside of the membrane because we have more positive ions coming in. It's becoming more positive. You'll get a little tiny bit of potassium that trickles out just because potassium tends to be kind of a leaky ion, but you're going to have way more sodium that comes in. And so when that happens, the inside of the membrane changes to be more positive and the outside of the membrane is now more negative because you became more positive on the inside due to more positive ions coming in. So this is what we call depolarization. Then, when you have the inside has enough positivity, it hits a threshold, and that causes your action potential. And so when you get your action potential, now what's going to happen is all along the membrane, you get lots of sodium-specific gates that open, and only sodium rushes in, and it starts to boom, 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 change the polarity. So you start getting this rush of positive, 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 and it keeps on going. As sodium gates continue to open, more positivity comes in, boom, 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 and it changes the inside of the membrane to positive. So that is what we call an action potential, and it's what is called an all or none response. So once you pull that trigger and those sodium gates open, boom, it's going to go all along through the membrane. Then we have to have a way to repolarize that membrane, so basically reset it back to the rest. And so now what happens is that sodium gates are going to close, and now we get a mass of potassium gates that open. And so what happens now is now you get this rush of potassium leaving inside the membrane and going outside the membrane. And because potassium is positive, what it does is it creates the outside to become positive again. So it restores the positivity on the outside because potassium is positive. So that's what's called repolarization. The rush of potassium ions leaving, that means the outside becomes positive, and then we're going to restore the outside to positive, and now the inside is going to go back to negative because now we had so many potassium ions that went out, lots of positivity out here relative to what is now inside, so the inside becomes more negative, the outside becomes more positive. And then what we have, because of potassium overshooting, basically you have more potassium that ends up leaving and going to the outside of the membrane. Now we have to restore the resting membrane potential. And what that means is that we have this mechanism that is called the sodium-potassium pump, and it's very, very important that you remember this. The sodium-potassium pump is what will allow the membrane to come back to the right voltage, the right amount of signaling so that we can stimulate it again. And how it does this is that it will pump in 2 potassiums and then pump out 3 sodiums. So sodium that was in here now starts getting pumped back out, right? And then the potassium that was out here starts getting pumped back in, and then we'll restore the membrane potential. So when you look at it as a whole, right, here we are at rest, So more sodium on the outside, less in here, and then we have more potassium on the inside, but the proportion of what we have in positive charges on the inside relative to the larger amount on the outside makes the outside more positive, the inside more negative. Now we stimulate the cell. So we're going to start depolarization. So what starts to happen is sodium starts coming in, and once that sodium starts coming in and depolarization happens, that triggers the action potential, which causes a bunch of sodium gates to open, and boom, 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 boom. It rushes in a lot more positivity, and that changes and continues to change the inside of the membrane to positive, and the outside thereby becomes negative. Then we have to repolarize. So now what we do is we are going to close the sodium gates, open the potassium gates, potassium rushes out, the outside of the membrane becomes positive again because of the positivity of the potassium, and now there's less positivity on the inside, so that means the inside becomes negative, the outside becomes more positive. Then to restore the potential, now we have our pump, and we pump in 2 potassiums and then pump back out the sodiums, and the ratio is 3 to 2. So for every 2 potassium that we pump in, we pump out 3 sodiums, and we restore our resting membrane potential. So when you look at this in terms of an electrical activity, we can actually measure it. And so what we have is the threshold potentials, right? So now we have what's called the membrane potential. It's measured in millivolts. So what we have here, this is your resting membrane potential. So now we have a neurotransmitter land on that receptor that causes that chemical gate to open. So what's going to start happening is we have sodium coming in. So sodium is coming in, and then it continues to come in. We have our depolarization happen, and then boom, we have all these sodium gates that open up that cause this major positivity to occur on the inside. That is our action potential. Then when we're done doing whatever we're doing on the membrane, now we close the sodium gates, potassium gates open, and now what happens is the inside of the membrane becomes negative. So that's why you see this happening, right? Inside of the membrane becomes negative, potassium is leaving, going to the outside, outside is becoming positive, inside becomes negative, and then what happens is we have kind of this extra negativity that happens inside because too much potassium leaves. And so this right here is called hyperpolarization. So it means that more potassium left. So then we fix that by having our sodium potassium pump back in, 2 potassiums, pump out 3 sodiums, and then we'll restore the resting membrane potential. And so what you're seeing here is the actual electrical activity of what happens with the neuron. All right? I know that it kind of looks like an EKG, so what you see on an EKG is actual electrical activity of what takes place in the heart. Right? What you're seeing here is just electrical activity of what is occurring within an actual neuron. And then what you're seeing up here with what's called absolute refractory period and relative refractory period, you'll see this again when we talk about the heart. Absolute refractory, you'll see, starts right here where you get that depolarization, action potential, and then the repolarization happening. So what that means is that you cannot cause an action potential or stimulate a cell that's already been stimulated. You can't stack because it's already happening. All right? It's already going. So once we get here, once that resting membrane potential, right, we had the outside become positive, inside become negative, we're trying to restore that right after repolarization happens. And during hyperpolarization, this is what is called the relative refractory. And what that means is if you had enough stimulus, you could, right here, generate another depolarization action potential to happen, right? Ideally, you want it to happen here because it's much easier to stimulate the membrane when it's at rest. But during this hyperpolarization, it's very difficult to stimulate the membrane again of whatever that neuron is touching, whether it's a muscle, whether it's a gland, whether it's another neuron. But you still can during relative refractory. You cannot during absolute. So during absolute refractory, think of it as absolutely no way can you stimulate the cell. Relative refractory means, well, relatively you could have a strong enough stimulus to stimulate that cell. So that's what that refers to. So in terms of our signal transmission, so let's look at it in small steps. All right, so here's our axon. Here's our chemicals, right? Here's our synaptic cleft, and then here is the membrane of the cell that we're going to stimulate. On that membrane, you have receptors. So what's going to happen is you're going to have the signal coming down the axon of this neuron, and now we're going to cause changes to happen here. And what happens, what's really cool, is that we actually have little calcium gates on the ends of these little axon terminals that trigger the release of the neurotransmitter. So the neurotransmitter gets told, hey, release the neurotransmitter, or the axon terminal gets told, release the neurotransmitter. Neurotransmitter gets released, and now what's going to happen is that neurotransmitter is going to attach to that receptor. If this chemical is a stimulatory chemical, that means that now these gates are going to open. So we attach, chemical causes the opening of those gates, and now we're going to be stimulating the membrane. All right, so it's really important that this neurotransmitter cross the synaptic cleft and bind to its little gate, its chemical gate, or what's called a ligand gate. Once that neurotransmitter attaches, remember we're going to have sodium that's going to start trickling in. And when that sodium trickles in, what it's doing is now changing the inside of this membrane to becoming more positive. And we want enough of these little receptors here to receive that stimulatory neurotransmitter so that depolarization takes place. Once we're done with everything and the signal is done, so what that means is that now that neurotransmitter is going to come off. So that comes off, the gate closes, and it also triggers the sodium gates to close. Now what we're going to have is repolarization. And then we get potassium gates to open, potassium goes out and repolarizes that membrane, and then our pump comes in and restores the membrane potential. So the key thing in continuing the stimulation of a cell has to do with our little neurotransmitter staying on here. So as long as this is here, this cell will continue to be stimulated. Once it comes off, that means party's over, and we're going to have sodium-specific channels that are going to close, that are down the membrane, and then potassium gates open, and then boom. And then what also occurs is that the neurotransmitter actually has an enzyme that is specific for that neurotransmitter and basically cleans it up. So you can kind of think of these enzymes as little vacuum cleaners. Then in terms of how our nervous system is protected, so I'm going to go through three things. We've already talked a little bit about the astrocytes, but we have membranes that are the meninges, and then we have the cerebral spinal fluid. So the meninges are made up of three membranes, all right? The dura mater is the outermost layer, and it kind of folds into the brain and forms these things that are called the falx cerebri and the tentorium cerebelli. These are just folding in, and all of this dura mater is going to be under what's called the periosteum, which is that outer layer of bone. The middle layer of the meninges is called the arachnoid layer, and it's called that because it looks like spider webs, and this is going to be where you actually have blood vessels that are passing through. And then the pia mater is the membrane that is right on the brain tissue. So here's our bone, and then what you're seeing here is your spongy bone. So you have periosteum here, and then we have periosteum out here. And then we have our dura mater, and so this is what you're seeing here. Below that is the arachnoid mater, and then right on the actual brain itself is where we have the pia mater. And the arachnoid mater, again, remember, has the blood vessels passing through. The other thing that's really cool is that within the arachnoid mater, what happens is that we have kind of like these little eyedroppers, and what they do is they actually allow for any excess fluid that is being produced by the CSF and all that. It'll actually go in to the region where you have this venous circulation. These are parts of the venous circulation, the veins that we have passing through our brain, that then will dump excess cerebral spinal fluid into that and then take away the excess. So it's pretty cool. So this is what it looks like for real. So here's our skull, obviously hair and all that. And then the dura mater is this stuff right here. It looks like a kind of like a Ziploc bag, freezer Ziploc bag. Then beneath that, just underneath this, you have this spidery web looking stuff that is going to be where the arachnoid mater is. So all through here, all right, you'll have all of this arachnoid mater. And then actually on the brain itself, the shiny stuff, that is going to be the pia mater that you would see. So this isn't actually a true brain. So this is the cerebrum of the brain, cerebellum, and then what you're seeing right here, this is part of the brainstem, and then this right here is all part of the spinal cord. And then these are the little spinal nerves that you see. Then your cerebral spinal fluid, why it's important is that it provides the buoyancy for the brain, and it also helps with the pH and the electrolyte balance. These are made, the CSF is actually made in the ventricles. So when you look at the diagram of what you're seeing here, this is a frontal view of the brain. So we have two lateral ventricles, one on the right of the brain and one on the left. And then we have a third ventricle more posterior, and then we have a fourth ventricle that sits just anterior to that. Here's your cerebellum, and then here is your brainstem. And so this is where you have the fluid that forms the CSF. When we look at it laterally, right, here's a lateral ventricle, here's a lateral ventricle, here's your third ventricle, and then you're going to have a little tube that is called the cerebral aqueduct that then brings the fluid down to the fourth ventricle. And why this is important is that the fourth ventricle is where the fluid is then going to go and exit out what's called a lateral aperture, and then the fluid will go around the brain. We also have what's called the median aperture that goes directly into this little piece that's called a central canal within the middle core of our spinal cord, and that is how the spinal cord gets the cerebral spinal fluid traveling down. When you look at the actual buoyancy of the brain, you can see here that as that fluid comes down and exits out of through its little canals, through the lateral apertures, it then fills and then provides the fluid that the brain itself is now going to sit in. So our brain is actually sitting in a fluid environment to protect it. And then we have the little central canal here through the central aperture, median aperture, and then that leads down into the central canal for the actual spinal cord. And then down when it gets to the bottom, it kind of comes out and then surrounds the actual cord. And so this is why when you have an infection in the brain, whether it's bacterial or viral, when something's going on, you can actually do a spinal tap, because the remnants and the clues of what's happening in the brain are going to be in the CSF. And so we can do a spinal tap to actually measure what is in that cerebral spinal fluid. Then the excess CSF that gets produced will then get dumped in through these little arachnoid villi into the venous circulation of the actual brain. And then that gets dumped into regular circulation, and then we get rid of the excess. Then our astrocytes, we talked about them again, but I wanted to bring them up because they're also part of the protection of the brain. And then these again are the little big paws that come off the body, and then they surround the capillaries of the brain, and then you'll have also pieces of them that surround the axons that are coming off neurons. And so they help maintain the polarity of what happens within an axon. Then in terms of the actual brain anatomy, so we're going to go through what the brain regions do and just in general describe what the lobes are, and then what the brainstem does, and then get into the actual cranial nerves. So this whole piece here that you see where these crinkles are, this is a hemisphere. So we have two hemispheres, left and a right, and this is the cerebrum. Then we have this portion here that's in purple that's called the diencephalon, and then we have the brainstem, and then the cerebellum. So these are your primary brain regions of a brain. The cerebrum itself is made up of two hemispheres, so you're going to have the left and the right, and down the middle we have what's called the longitudinal fissures. This is what separates the left brain from the right brain. Then within our actual brain, these things that look like worms, right, or like gummy worms is kind of what I think of. These are called a gyrus, right? All of these together are gyri, that would be plural. And so each of these make up part of that cerebrum, and they're put together, right? So when they're together like this, that is what you're looking at in terms of the cerebrum. The brain itself, down the middle, right, between the frontal lobe and the parietal lobe, has this piece here that is called the central sulcus. So it's labeled right here. So this is your central sulcus. And why that central sulcus is so important is that it actually separates the frontal lobe of the brain from the parietal lobe. And why that matters is that your frontal lobe is your motor, and the parietal lobe is the sensory portion of the brain. Then what we have here, obviously, are the lobes. So here's your frontal lobe, and then we have the parietal lobe, separated by the central sulcus. The frontal lobe, what you need to remember about this is where your motor control, this is where your personality is housed, where you make your judgment, and a lot of reasoning is taking place. So this is why frontal lobe injuries can cause so many problems with individuals. The parietal lobe, this is where the sensory information is going to be coming in, and then it's separated again by that central sulcus. Then what we have here is the temporal lobe, what's called the insula, and then the occipital lobe. So your temporal lobe, this portion, why that matters and why it's so important is because it's where we have our smell, our hearing, speech. So this is where you're going to have the ability to actually talk, right? Speech is received, so we'll get to that in a little bit. And then a huge point of memory, obviously, with what it is that you're going to hear and then what you smell. Then the insula, this portion here, this ties into our gut. So this is where we have a lot of the information that comes in from, if we're feeling full and where it's located and all that, and our center for hunger and all those areas. And then our occipital lobe, this is where our vision is. So we actually do see with the back of our heads. So you kind of have eyes in the back of your head, but that's your occipital lobe back here. Then we have the special senses, so in terms of where they're wrapped up. So here you see a little bit more color to our actual brain within the lobes themselves. And our visual area within the occipital lobe is broken up into the visual association area and then what's called the primary visual cortex. So we see it here and we make sense of it here. Then the auditory area is really right here in the temporal lobe, and this is where we actually hear, right, where the sound comes in. And if you're able to hear, this is where you hear. And then right here, this little area that's called Wernicke, we'll see that in a little bit, this is where you actually make sense of what you heard, right? This is where you understand words. And then the olfactory area, the olfactory association area is going to be here, and this is where our olfactory bulbs are located that are receiving the chemical smells from what is in our environment, and this is where that is. Then in terms of what I talked about earlier with white matter and gray matter, so when we take a brain and we slice it, so this is an actual human brain, you see the areas that are white. These are all regions that are myelinated. So you can see that the majority of the brain actually has axons passing through it that have a significant amount of myelin. So those are very fast signals. Then you'll see that the outer regions that are more gray, and there are some clusters of gray within here, these are all the gray matter. Right? This is called the cortex, the outermost portion of our cerebrum. This is where we also process a lot of information, and a lot of thinking takes place here, but this is the gray matter. It's about 10% of the volume of that cerebrum. And then within the brain, when I talked about different nuclei, you'll see these little clusters of gray matter. That's where the nuclei are located. So that is what you look at in terms of what white matter, gray matter, and then what is our cortex, and then where we have the nuclei located. Here you're looking at a sagittal view of the brain, so you can actually see from the side. So this is the medial portion of the right side of the brain. So out here is our cerebrum. The outermost portion is going to be the cortex. And then here in the middle, this piece that you see is called our corpus callosum, and I'll talk about that in a little bit. And then here is what one of the lateral ventricles looks like. Here you can see the superior view of the lateral ventricles. And then this piece right here, this is our brainstem, and then right here is our cerebellum. Right back here is where the third ventricle is located, and then as you pass through here, this is where our fourth ventricle is. Then in terms of the special senses, again, when we talked about vision and when we talked about auditory and the ability to talk, all right, those are going to be highlighted here in this area. So here is our Wernicke, where we receive the information to think and talk right here, right here. This is our gustatory cortex, where you actually feel stuff related to taste. And then this is our Broca area, where we actually form the words and use the muscles to actually speak. Then what's called the primary somatic sensory area. So I talked about the parietal lobe being our sensory cortex. This right here, this what's called the postcentral gyrus, this is where we receive information from our skin, from our muscle, from our tendons. This is where we're actually feeling those areas, all right? And what this is called in terms of the distribution, it's like a spatial map of our body parts, right? I'll show you that in a sec. Then what's called the primary motor area, or the precentral gyrus, is this piece here of the frontal lobe. And this is where we're going to be sending the signals to the muscles so that we actually conduct and do these motor activities based on what we felt in those body parts. Like say it was your arm or your face or your leg that you felt over here, then that signal transcends over to this side to then send the signal on down. So when you look at it as a whole, this is basically how they're mapped out. So here is that precentral gyrus, which is the motor cortex, and then over here in the blue is the postcentral gyrus, which is the sensory, right? And so what you're seeing here, for example, is here's your hand, right? So this whole area of the surface, when you actually are going to move your thumb, move your fingers, those signals are originating here to go down, cross over to the opposite side, and then go to the muscles and the neurons that are going to be sending the signal to those muscles. Over here on this side, where you felt that feeling on your thumb or what you touched with your fingers, right? That information came in here, but you notice that this piece here where the hand is and this piece here where the hand is are actually exactly in the same spot, just on opposite sides of that central sulcus. So these things are called a homunculus, right? And it's just a sensory map and a motor map of where that information comes in and where the information goes out. Then the Wernicke area, which was right here, this is again where you're going to receive the sound. So this is, remember, your auditory association area. This is where you're going to make sense of the words that you hear, the words that you read, all these things, you make sense out of it right here. Then when you want to say it, this is going to be right here. So this is your Wernicke or your Broca area. So Wernicke listens, Broca speaks. It's the easy way to remember who does what. This would be a kind of fun question that they could potentially ask you on registry in terms of what Wernicke and Broca are, because when you start learning about problems that occur with psychological diseases, so for example, if you have schizophrenia, right? People that have schizophrenia think voices are talking to them, right? When that happens, they are actually hearing things, stuff is going on right here in that Wernicke, right? Like you and I know that there aren't voices talking to somebody, but they're actually hearing it here. And then when you're going to make sense, you're going to talk, right? Now that's going to come from Broca, which is located here proximal to where that region is in that motor cortex where you're actually going to speak the words. And so if you look at where the Broca area is relative to where the actual speech, where you're going to voice and use those muscles, look at where it is. It's on that section of that motor homunculus in that precentral gyrus here, and then here is your Broca area. So that is what those very specific areas for words and speech are located. Then our primary motor cortex, again, here. Sensory is going to be here. Broca, we speak. Wernicke, we listen. Then in terms of how we communicate and how the signals are going to pass through within the brain and then from side to side and then up and down the spinal cord, we have very specific communication fibers, right, or communication neurons. So you know that we have two hemispheres, right, a left and a right. Then we also have the tracks that are going to run from our brain down to our spinal cord and then on out. So what you're seeing here is a diagram that just depicts what the fibers are. So we're going to have what are called commissural fibers, which go from left to right, right to left. Then we're going to have what are called association fibers, which are only on one side. So they only communicate within one hemisphere. They stay on their own side. They only associate with their own side. And then what we have is going to be the tracks that are called the projection tracks that run up and down. And then these are vertical fibers that go from the brain down through the spinal cord. All right, so to summarize, commissural fibers go left to right, right to left. Association fibers stay on their own hemisphere. They only communicate within the left or they only communicate within the right. And then projection fibers go from the brain down to the cord and then from the cord up to the brain. So corpus callosum, again, I want to make sure that you guys remember what this is. This is that very thick white matter that connects the two hemispheres, and that is where you're going to have the thickest fibers, the more myelinated region of those commissural tracts that allow the left and the right to communicate. Then remember our association fibers, those are going to be within the hemisphere. And then the projection tracts, these are going to be from the cerebrum portion of the brain down through the spinal cord. Then the basal nuclei, I talked about these a little bit before, but why I want to bring them up is that these are absolutely critical in terms of what they're going to do with voluntary motor activity. And what that means is that they are very important in helping make sense out of and smooth out movement that is then being coordinated from your cerebellum to the voluntary portion of that frontal lobe that is then going to send those signals down to the skeletal muscle. So these basal nuclei are absolutely critical for that. And they are very closely located to a portion in our midbrain, which is part of our brainstem, where we have a region that is damaged when somebody has Parkinson's. And so this is why sometimes you'll see these tremors that occur with individuals that have Parkinson's, because the signal smoothing out is actually being damaged. Then the diencephalon, that is this big purple area. It's actually a region that has 3 subparts that are very important. We have the thalamus, the hypothalamus, and then what is called the epithalamus. And so what you see here on this portion, all of this right here, this is your third ventricle. And then the portion that surrounds that third ventricle that is in this piece here, this is where the thalamus is located. Then we have the hypothalamus, and then below, above that, over here in the little posterior region, this is where that pineal gland is going to be located. So this is kind of considered to be the epithalamus. In terms of what they do, the thalamus, very, very important in terms of being a relay station for the sensory signals that are going to be coming through and passing, that are going to travel up to the cortex when you're receiving information in that parietal lobe as far as sensation and what you're feeling and what you're making sense out of with that. Then what you see here, this is a sense of where that thalamus is located and then how it's going to radiate up here to that cerebral cortex. So the information is coming in. Then the hypothalamus is a very important region that has a whole bunch of little clusters of nuclei that actually regulate hormonal function. So it's kind of like the region in the brain that is going to direct the function of the pituitary gland and the hormones that are secreted from there. And in doing so, it also helps in terms of regulating body temperature, water balance, metabolism, reproduction, thirst, hunger. All these primary functions of what we need to keep us alive are regulated by that hypothalamus. And then the epithalamus, this is where the pineal gland is located. And what you want to remember about this is that the pineal gland secretes melatonin, which is a very important hormone that helps us with sleep, and it helps with regulation of day and night. So our circadian rhythm is very important here in this area. And then within the epithalamus is also the portion of where that ventricle, the third ventricle, is located that will help produce some of that CSF. So we've got the thalamus, which is going to be in this area here. Here's our hypothalamus that regulates directly. This is your pituitary gland here. We'll talk a lot more about that when we get to the endocrine system. And then back here is the epithalamus, and this is where that region is of where the pineal gland is located. Then our limbic system, why this is important is that you'll see all of this purple stuff in terms of the sagittal view of the brain. All of this is regulating our emotions. So it regulates how we are receiving the information, right? So how are we going to process it? How are we going to be remembering what we're feeling emotionally based on what that experience or stimuli is? And then how are we going to react to that? What are we going to do? So emotion is very much tied into what happens in our prefrontal cortex here, which is where our judgment, our personality, all of that is located. How that information is going to be communicated, right? Between, here's our corpus callosum, this big white piece, and then this thing that's called the cingulate gyrus surrounds that and helps integrate some of that memory, all right? And then here we have a hippocampus, and then here's the amygdala. So all of this is where that limbic region is located. So this is our big emotional center and a very, very important piece in terms of how we learn and what state you're in when you are learning, when you're integrating that information. So gratification center, obviously these are the sense of pleasure, so things that feel good, whether it's an actual feeling of being with somebody where you're bonding with them, or it's a feeling of, say, you took a medication and wow, you know, that made me feel good. So like caffeine, right? Caffeine is a stimulant, helps us feel good, helps us focus. That is kind of where that ties into that gratification centers. Aversion center, this is going to be the region where you're going to integrate those unpleasant memories, things that don't feel good, things that create fear, things that trigger a negative response. That's where that aversion center is located. And so the cingulate gyrus, that's this piece here. Lots of processing here that occurs in terms of thinking, working through problems when you're concentrating, when you're really trying to figure something out that is actually involving conflict or you're frustrated. And then how you're going to deal with that is going to get translated here to that piece of, okay, this is how I'm going to react. These are the things that I'm going to say. And so why I have this piece of the brain so that you could remember that where that limbic system is located and what is taking place in those regions is directly tied in also with what overlaps in terms of our auditory center, our Wernicke, right, where we integrate what we hear in terms of the voices and sounds, and then Broca, what are we going to say in terms of responding to what we felt. So that's why I wanted to show you that piece here. Then the hippocampus, that's this part right here. And the hippocampus has a lot of memory that is integrated there. It's a huge memory center where we've learned a lot of things and kept them. So within that, it's a piece, and what's kind of cool is that we actually have stem cells for our neurons there that we can actually rewire. So when individuals are going through some kind of trauma therapy, right, this is where we're able to really focus on those areas and where the therapists are able to tap into those regions and then help us basically reframe how it is that we experience that trauma. And so they're able to do that because of the plasticity that we know that now exists in that hippocampus. So that's what the hippocampus does. It's a big memory center, and it's able to modulate and change how it is that we are able to remember and feel those memories. Then the amygdala, that's this kind of teardrop thing. The amygdala is where that aversion center is, where it senses danger, elicits the fear response. Really, really important also in terms of memory. And big thing with that is when epinephrine is involved in turn with stress hormones like cortisol, and then how the amygdala responds, it's very, very sensitive to that. And so that is why you can have these like traumatic negative experiences that just get ingrained in your head, and there can be those triggers that cause that to happen. And so the amygdala, I always kind of thought of it as the angry amygdala, and then the hippocampus, I think of it as more like the happy hippocampus, where you can actually change things to make it less stressful and less traumatic. Then environmental stimuli, so again, how you integrate smells, how you integrate things you see, how you integrate sounds, that is all tied in to that limbic system as well. And that is also obviously relayed through the hypothalamus because we have a region there that also helps control how we react to stress. And so within that and the environmental stimuli, how we regulate our stress levels is going to then impact directly how it's going to affect our organs, right? So cardiac function, respiratory function, gut, huge with our gut. How do we react to that? And so is it going to be more of an acute response, or is it going to be a chronic stress response that is going to continue and then over time cause problems? So the limbic system is very much tied into that. So, and then how it interacts with this purple part here, which is our prefrontal cortex, this again then ties into how we're going to express ourselves, right? So how are you going to say things? What are you going to do? And that is this big piece that you're seeing here. The other thing that is, just want to point out, is this frontal lobe portion of our brain and this portion here with our prefrontal cortex and how we're going to express ourselves and how we're going to react. This isn't done maturing and developing until we're about 24, 25 years old. So this helps to explain, in a way, why it is that teenagers have such stupid ability to make stupid decisions. And so when you're looking at a teenager, you're like, God, what is wrong with you? Like, what made you think that was a good idea? Well, they're not done baking yet. And so that's this prefrontal cortex portion that causes that to occur. Same when you're having individuals who've had a frontal lobe injury or a frontal lobe dementia. What is happening is that this portion of the brain is actually loses its ability to actually do its job well because you've actually damaged that part of the brain through the brain injury. And that is then what causes for those changes to happen in terms of behavior, or if the person's personality changes in terms of, before they used to be really nice and then they just turn into complete assholes. So that can happen here because of what happens to the prefrontal cortex. All right, then moving to the back of the head, so where our cerebellum is located, this is almost like this little mini brain. but it's really, really important in terms of motor control, but then it also contains a lot of memory. And in terms of the memory, then also pieces in sometimes in terms of emotion. So in terms of the actual physical pieces of coordination and balance and agility, that is regulated by the actual cerebellum. And so this is where we have our timing for skeletal muscle activity, how we're going to coordinate body movements together. So when you're learning how to walk as a baby, right? You're learning how to take those steps, how to push yourself up. All those things are taking place in coordination with what happens with the cerebellum. And then how did that feel, right? It ties into what's going to take place in terms of the limbic system. Was it a good experience, bad experience? And then when you're, for example, learning how to turn left, right? You're learning how to drive, now you have to make a left turn into oncoming traffic. This is where you learn that timing, right? How are you going to do that? Or when you're learning how to catch a ball, how are you going to determine the timing in terms of the motor activity to catch that? That is what takes place with the cerebellum. Then our brainstem sits right before that diencephalon, and it's composed of the midbrain, which is depicted here in the orange, the pons, which is the lighter green, and then this region here, which is our medulla oblongata. The midbrain portion here ties in some of our cranial nerves that we're going to get into, and this is where we have our visual and auditory reflex centers. So for example, when you see something that is dangerous and it causes you to kind of like, boom, make a stop, that is where that reflex is coming from. When you hear something and you turn to look to see where that sound came from, that is a reflex that occurs, and it's actually mediated through this portion of the midbrain. Very important portion for that. Then the pons is this middle portion. So this is where we have part of our breathing center, but we're also going to have other very important cranial nerves that come from here that have to do with our ability to taste and feel sensation within our mouth. And then also our ability to be able to balance. So right here at the very bottom of the pons is where we have a nerve that is going to help us with hearing, and then also our equilibrium. So a very important region. And then the medulla oblongata, the real critical piece that I want you to remember about this is that this is the portion of the brainstem that actually connects to the spinal cord. So spinal cord is here, midbrain is here. And located proximally to that spinal cord is that portion of the medulla oblongata where we have our cardiac center that regulates our heart rate, the vasomotor center which regulates blood pressure, and the respiratory center that is going to regulate the frequency and the depth of our breathing. So really critical areas that help keep us alive. And this is why when you have spinal cord injuries that occur around C1, C2, C3, this is why they can directly impact the regions here that regulate our heart rate, our respiratory rate, and then also our blood pressure. And this is why those high cord injuries can end up killing somebody because of the inflammation that then occurs within that portion of the brainstem. When you have a brain herniation where you actually have the brainstem itself pushing through and going out through that large hole in our occipital bone, the foraminal valley, now you have that intracranial pressure up here that is occurring, pushing down on that brainstem, and then that's going to directly impact those survival centers for our heart, blood pressure, and breathing. And then what we also have here is what's called the emesis center, which is what triggers vomiting. So this is why when an individual in some cases has a brain injury, they're actually also going to vomit. That's going to be one of the signs that they show. Then the reticular formation, last little portion here that is just at the very top of our brainstem, right above the midbrain. This is also involved in terms of motor control of our visceral organs and our consciousness. So this reticular activating system, right, so here's the midbrain, it's right about here. And then this piece here is absolutely critical in terms of can you rise to consciousness, right? So things that when you wake up, when you go to sleep, when you're under anesthesia, this is the area that's inhibited. This is what they're targeting so that you won't remember and you won't feel. All right. And then obviously if it gets to the cortex, now it's coming to your consciousness. You're actually processing that information. So that is why obviously when you're having surgery or something that's going to be very painful, they'd like to tamp down and turn that off. Then our cranial nerves, these are now also part of the peripheral nervous system. We have 12 pairs, so 12 on the left, 12 on the right. And they are primarily going to serve things within our face, but we're also going to have some of them that are actually going to leave within the skull and leave the brain and then go on out. So what you're looking at here, this is an inferior view of the brain, and you have the brainstem. So this right here, this is part of the olfactory nerve, where the olfactory bulb, this one has now receptors that actually reach down into the nose. Right here, this is the optic nerve. So we have obviously coming from our eye, so this is what provides our vision. And you see that there's this little bridge here, it's called the optic chiasm, where you actually have fibers that cross and go to the opposite side. What you're seeing here, this is the opening of the carotid arteries. So this is where our blood supply for the central portion of the brain comes in. Then this is the third cranial nerve. It's called the oculomotor. And what it does is that it regulates motion of our eye, so the movement of our eyes, and then also our ability for our pupil to dilate, to actually constrict. So that's regulated by the third cranial nerve. The fourth right here, this is called the trochlear, and what it does is it also regulates eye movement. The fifth right here, this actually is called the trigeminal, and this one goes into the mouth and helps us with taste and then feeling within our gums. The sixth cranial nerve, this is called the abducens and helps regulate motion in our eye. The seventh, this is our facial nerve, and it has 5 branches that are going to go down into our face and regulate movement of our muscle, stimulation of our salivary glands and such. And then the eighth, this is the vestibulocochlear nerve. This is what helps provide our ability to hear and our equilibrium and our balance. Then the ninth and 10th are right in this area, and the ninth is the glossopharyngeal, which helps us with swallowing and blood pressure regulation. And then the 10th is our vagus nerve, which helps us with the majority of our parasympathetic functions, regulating our resting heart rate, resting blood pressure, and respiratory rate. And then the 11th, which you can't see very well on here, but it's going to be just right about there. That is where it's called the accessory nerve, and it allows us to move our shoulders. And then the 12th is the hypoglossal, and what this one does is it helps us in terms of movement of our tongue. So we'll go through these in detail, and here you can kind of see a diagram in terms of where you have them. So when you look at it, in the midbrain is where you're going to have the nerve passing through for the olfactory, which is cranial nerve number 1, the optic nerve, cranial nerve number 2, the third, cranial nerve number 3, and then 3 is oculomotor, and then 4th is trochlear. Then the pons, where you have the 5th through the 8th cranial nerves. So the 5th is the trigeminal, the 6th is the abducens, 7th is facial, 8th is vestibulocochlear, and then within the medulla oblongata, this is where we have the 9th cranial nerve, glossopharyngeal, 10th the vagus, 11th the accessory, and then 12th the hypoglossal. So a lot of mnemonics, things have been made up to try and remember, okay, what are all these cranial nerves? So this one of O-O-O-2, touch and feel very green vegetables, right? Just a stupid way to remember the order of the actual cranial nerves. The reason why I'm making such a big deal of these is that you will very likely get at least 1 or 2 questions of these on the National Registry. And these are easy money points that you can get right when they're asking about things related to anatomy. So let's go through them in detail. So what we have we're going to have 3 sensory-only cranial nerves. So the first 2, which means that they don't provide any motor function whatsoever. They only sense things. So olfactory obviously is your sense of smell. So that is your first. So cranial nerve number 1 is the olfactory nerve. You have 1 nose, easy way to remember it. The optic nerve, you have 2 eyes. So cranial nerve number 2 is the optic nerve. This is your ability to see. So what it is that your retina is where you receive all those light waves that are coming in, and then they go to the posterior portion on the retina and then go into the optic nerve that will then have some fibers that cross over to the opposite side here at this optic chiasm, and then some of them will stay on the same side and then go directly to the thalamus. So we'll talk more about those when we get into how we see and what our vision is in the special senses chapter. So that's your optic nerve. Then when you look at and in terms of what's also cool about the cranial nerves is you can actually test these with your patients if you're suspecting that somebody's having some kind of stroke. Olfactory, you just ask them, if they can smell things or if they've lost their sense of smell. For optic, obviously, if they can see an object, or you can hold up your fingers and ask them how many fingers do you see, right? That's an easy way to test that. And then the third sensory-only nerve is actually cranial nerve number 8, and it's 2 nerves merged together. That's why it's called vestibulocochlear. The vestibular portion of it is your equilibrium. right? So that is what you see over here in the inner portion of the ear. These are called the semicircular canals. And in here is where you have receptors that tell your body and your head, what position am I in? Am I static, staying still? Is my head down? Is it up? Is it to the side? Or am I moving? Am I spinning? And that is where all that information comes in and goes into the vestibular portion of the nerve. Then your sense of hearing is right here, and that goes into the cochlear portion of the nerve. So that is why it's called vestibulocochlear. So the way that you can test these is you can have a patient repeat a phrase that you say, right, for the hearing. How do they sound? Are they able to say the words? And do the words come out right? And then with equilibrium, if the patient is able to stand, you can have them stand on one leg, or you can ask them if they feel dizzy. So that is what these vestibulocochlear nerve does. Then we have motor-only cranial nerves. So the third cranial nerve, the oculomotor, what it does is that it's going to affect the pupils in terms of their size, their shape, and their reactivity. So when you're looking at the pupil, right, are they pinprick, or are they really dilated, or are they normal? Are they pearl? That is what the oculomotor nerve test would be. The reactivity obviously is very important because when you shine a light, what you have is if the oculomotor nerve is working and activating, it's going to constrict that pupil. So that's what it means. It's reactive, right? You take that light away, now the pupil should dilate back to normal. Then what it also does is it innervates the branches of the lateral, medial, and inferior branch of these rectus muscles that move the eye. And what's important about these and the way you can test it is you can have the patient follow your finger and you can move it left and right. And so if the patient is able to, as you move your finger to the left, the right eye should be able to move medially. That is what that oculomotor nerve is going to do. When you tell them to look up or look straight down, that is also an oculomotor nerve activity for those muscles. The trochlear nerve regulates eye movement as well, but this one only innervates what's called the superior oblique muscle, and what it does is it actually allows you to move your eye inferiorly and to an angle. So down and then an angle. So you would just do an angular movement with your finger and ask for the patient to follow it. Then the sixth cranial nerve, the abducens, this one innervates what's called the lateral rectus muscle, and this one allows for eye movement laterally. So when you hold your finger up and you have the patient in the transverse plane, just look to the left and look to the right. The right eye should move to the right, and then the left eye should move to the left. And so that is what you're seeing in terms of, does the abducens do its thing and is it working? Then the accessory nerve actually innervates the sternocleidomastoid muscle of the neck and the trapezius muscle in the upper portion of the back. So what you have for your patient do is to rotate the head left and to the right, and then for the trapezius, just have the patient shrug their shoulders up and then down. So that's what the accessory cranial nerve number 11 does. And then we have the mixed nerves. Mixed means that they have sensory components and motor components. So the sensory nerves for the trigeminal cranial nerve number 5, what they are is that they're going to have information coming in terms of being able to feel touch or temperature on the forehead, on the nose, above here, pieces of the eyelid, on the side where the temple is, and then going into the maxilla where the gums, and then under here with the tongue in terms of taste, and then also down under with what you feel within the gums and the teeth and the mandible. So that's the sensory portion. The motor piece is going to go directly into the corneal reflex, and then your ability to open and close your mouth. So opening the mouth, obviously, and then closing is going to innervate directly into this very important muscle called the masseter, and then also down below where you have the muscles that are called the pterygoid muscles, which help with the movement also of moving backward and then being able to swallow as well. So these are all really, really important muscles within the lower portion of our jaw. Then the facial nerve has 5 branches. Back up here, easy way too to remember, trigeminal, tri means 3, so it's got 3 branches. And these are those points of sensation on the face that you would feel. And then the motor part ties in directly to the innervating your eyelid, so your ability to blink, and then your ability to move muscles of the face. So this is why when some individuals have Bell's palsy, which affects directly the facial nerve, you'll see some of that drooping on the actual face. And then what it also does is it innervates with the motor part, tearing, the ability to tear, so it's going to have some parasympathetic branches to it, and then going down into the salivary glands, it helps in terms of secretion of saliva. And then number 12, the hypoglossal nerve innervates directly into the tongue. So you get a little bit of sensation there, but also that motor portion of also innervating the muscles that will help you with actual swallowing with the tongue. Then number 9, the glossopharyngeal, very, very important with this one in terms of also taste, but real critical in terms of blood pressure regulation, and then also with swallowing because of how it impacts the ability of the larynx. And so what you're seeing here, this is, you're going to have sensory fibers that are going to be going to these things that are called the carotid sinus of the actual carotid arteries that measure pressure. Because your carotid arteries, as you know, are going to supply blood into the brain, and you don't want the pressure of the blood as it's entering the brain to be very, very high. So it regulates how much blood flow is actually passing through those arteries. So that's your glossopharyngeal nerve. So you can ask your patient to swallow, and that is going to give you indication if the glossopharyngeal is working. Same with your hypoglossal. You can ask the patient, is it swallowing with your facial? You can ask your patient to blink and then also to smile. And then the vagus nerve, really super important. This is basically our parasympathetic function of our body. So that is why I always think of this as Vegas, like Las Vegas. If you cut this nerve or you damage this nerve, it's going to end the party. And so that's cranial nerve number 10. So the sensory portions, you're going to have signals coming in that are going to come from the pharynx, the larynx. So the swallowing is real critical for this. Same thing in terms of if anything goes into the larynx, you're going to have a glottic reflex, which is going to cause you to then cough and or do something to cause whatever is trying to get down and block the airway to react. And then it also has the motor fibers that go directly down into the lung to signal the ability to breathe, right? Your respiratory rate is going to have direct impact in terms of what the vagus nerve is doing. And then also our heart rate regulation, really, really critical for that. And then it also stimulates, obviously, digestion, and then what happens with our kidneys in terms of urine formation. So this is huge for our parasympathetic function. So branches of our cranial nerves that are going to have parasympathetic function are going to be our third, the oculomotor, our oculomotor nerve, the facial nerve, number 7, and then glossopharyngeal, number 9, and then number 10, the vagus nerve. And when we get to the parasympathetic system, you're going to hear me talk about that again. And then I also have these tables included that go through and summarize where the nerve is actually located, what its function is, and then what would the test be that you can actually do as a medic. And then here's 3 and 4, so I'll just have you guys can read through these on your own and help you in terms of the memorization. And I've also included a table for you to go through and also fill it out as just to fill in the blank so that you can start learning what the cranial nerves are, their number, where they're located, and then what they do. So here's 9 and 10, and then obviously 11 and 12. All right, and now we're going to get into what the spinal cord does and how it functions. All right, so the spinal cord, what we have is going to be obviously our cranial nerves that are going to come off of it. But the spinal cord itself, without the spinal nerve, is part of the central nervous system. So we have 31 pairs of these spinal nerves. The spinal cord also has gray matter and white matter. And again, remember that gray matter means that it's not myelinated. And then within the actual spinal cord, we have these things that are called horns. And the horns are part of that gray matter. So when you look at it, these are the horns. So this is your dorsal horn, this is the lateral horn, and then this portion here is going to be the ventral horn. And why these horns matter is because signals that are going to be coming in to the spinal cord from the fibers, the sensory fibers that come in through the dorsal horn. Then motor signals are going to originate here in the ventral horn and then exit out. So what happens if you have a signal coming down from your brain that is a motor, right? It's going to move a skeletal muscle. It'll connect to another neuron here. This is where the little soma of that neuron is. And then that neuron, the axon, will leave through the anterior portion of the cord and then go on out. The lateral horn here, this is where the origin is for what's called the preganglionic neuron in your sympathetic system. So when we get to the sympathetic system, we'll be talking a lot more about this lateral horn. So the horns, remember, are just the pointy parts in the gray matter of the spinal cord, which is located at the core. This stuff that you see out here that surrounds the gray matter, this is the white matter, and those are called columns. So you have dorsal columns, lateral columns, and then anterior columns. The white matter, like I said, are dorsal, lateral, and ventral. And so what's important about these is that we have actual fibers, tracks, running up and down. And so remember that sensory is going to be called afferent. So that means that these afferent tracks are going to be ascending tracks, which means they're conducting signals that are going up to the brain. And then the signals that are coming from the brain down are descending signals, and those are going to be the ones that are going to the skeletal muscle, and they're called descending. So what we have here's our dorsal column, lateral column, and ventral column. The way that the tracks work, and why I love this image from, you guys don't have this one in your textbook, but I always like showing you this. So here's our cord, right? Here's our gray matter. So what will happen is that we'll have a signal coming in. So that's what you see here, signal's coming in. It's going to enter through the dorsal horn, and then it's going to synapse, so it's going to connect to an actual neuron, right, that is going to be passing through. So it goes through the dorsal column here, goes up, and then here in the medulla oblongata is where it's going to synapse with another neuron that will then cause it to cross to the other side and then travel up into the brain. Here at the thalamus, it synapses with another neuron that then sends the signal to the cortex. So for example, when you actually, this portion of the brain up here, all right, so here is the face down here, is this part where the hand is located. So we have sensation on our hand. So we feel that coming in, right? It's coming in through the sensory. It's going to come in through the dorsal horn, go up through the dorsal column, synapse in the medulla oblongata, continue its track, go to the thalamus, and then boom, you feel it up here on your hand. And so the ascending tracks that come in actually come in, and it's a 3-neuron sequence. So you have a little relay of little neurons in a row that are going to help you feel. But what I want to get the point across is that the ascending tracks coming in are going up. So what you're seeing here, these tracks that are ascending that you see here in red, these are also found on the opposite side of the cord. And these green ones that are the descending tracks are also found on the other side of the cord. It's just that in textbooks what they do is that in order for students, when they're having to memorize these, for example, in the higher level anatomy and physiology, you have to memorize all these tracks. They show it for simplicity so that you can learn the location of the ascending, the location of the descending. But again, these red ones are also on this side, and then the green ones are also on the right side. And then the descending tracks are going to be the motor tracks. So these are the signals coming down, and these are primarily coming from regions in the brain that are then going to come down and hit the portions that would be more the motor. And so then these descending tracks would then connect to the neuron that is then going to exit out through the ventral root here and then go on to the skeletal muscle. So spinal cord tracks, just remember, ascending are sensory, descending are going to be motor. And where these tracks run are going to be on the columns of the spinal cord. Then when we talk about these things that are called ganglia, remember, are just little swellings in a nerve. And what's important to remember about that is that these little swellings in a nerve, for example, this one's called the dorsal root ganglion, they actually contain little cell bodies in them. They have somas, they have portions of a neuron that is passing through there. So in this case, this little guy here, the dorsal root ganglion, contains the soma for a sensory neuron that is going to be bringing the sensation in through this dorsal root into that dorsal horn, and then that will continue into an ascending tract that will go up into the brain, or in some cases it connects to an interneuron that will then send a signal immediately out a motor neuron in the case of a spinal cord reflex. So ventral root and dorsal root, what I want to explain about these. Dorsal root here, this is sensory only. Ventral root is motor only. So what happens is anything that is a motor signal that is exiting the cord is always going to go out the ventral root. Anything that is a sensory signal coming in is going to come in through the dorsal root. So the dorsal side of the cord is going to be the portion that brings in sensory information, and the ventral root of the cord, anterior portion coming off the ventral horn, this is going to be motor information or efferent signals that are also going out. What's also cool is that as these signals go out the ventral root, because I told you earlier that the lateral horn is for the sympathetic neuron to come out, what is going to happen is it also exits, it shares this portion of the ventral root with the somatic motor or the motor control, voluntary control for your skeletal muscle. So all you have to remember with this is dorsal is sensory, ventral is motor. Again, dorsal is sensory, ventral is motor. Dorsal, remember, is also going to be where the afferent sensory signals come in. So remember that the word afferent is always coming in to the central, and this is why the dorsal root here, we call it sensory or afferent as well. So any questions on this, please let me know, because this can sometimes confuse people. Then our dorsal root ganglion, like I said, this is a little swelling in that dorsal root. So what is happening is you're going to have sensory information coming in, and what we have here is what is called a unipolar neuron. So as a signal comes in, right, it's going to come in, processing is going to happen in this little soma, the signal continues in to that dorsal horn, and then the message will go up to the brain through an ascending tract, which is an afferent tract, sensory tract. And then the information will come back down through that signal that would then obviously exit out and then go into the actual muscle tissue. So, but the dorsal root, remember, is going to bring in the information. And then a ganglion is just a little swelling in a nerve, and within that little swelling you contain little somas or little cell bodies where information is being processed from that sensory neuron that is coming in. So that's your dorsal root ganglion. Then what a nerve looks like is very similar to what skeletal muscle looks like in terms of the wrappings. All right, so this right here, this is one actual axon, and then what you're looking at with the hole, this is basically a bundle of nerves. So if you think of this as Twizzlers, all right, this whole thing, all right, the outer wrapping, okay, it's called the epineurium. Think of this as the container that holds that whole big bunch of Twizzlers that you bought at Costco. Then each little bundle here is surrounded by this stuff that's called perineurium, and this would be like your hand if you're holding a little group, a little bundle of Twizzlers, right? Because each one of these is a Twizzler. And then each one of these little guys, this is an axon. So each Twizzler is an actual axon. And then in some cases, they're going to be myelinated, right? To help protect them and help the signal go faster. So that is what, when we refer to nerves, right, they are in bundles. And so they're going to have wrappings that always protect them. And so that's why I wanted to show you this. And then you'll always have a really, really good blood supply going to nerves. Then what we have with mixed nerves, this just means that running along here, you will have nerves that are sensory, and then you'll have nerves that are also motor, and they're running side by side. Sensory nerves, again, are your afferent. They send the signal to the central nervous system. Motor are going to carry the signal away. And then with our spinal cord, we talked about the cranial nerves already. Now we're going to talk about the spinal cord nerves. We have 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and then 1 down here at the very bottom, which is the coccygeal. So an easy way to remember this is breakfast at 8, lunch at 12, dinner at 5, you get 5 snacks, and 1 cocktail or 1 cookie, depending on your preference. That's an easy way to remember these. And then you have 8 cervical nerves, because the first cervical nerve actually is located between the occipital bone and the first cervical vertebrae. The second cervical nerve is located between the first cervical vertebrae and the second cervical vertebrae. So that's how then they're numbered based on the vertebrae that they're located just above. So that's how we number these. When you look at just a little more close up in terms of, okay, where are they going? All right, what we have is in the cervical region, the 8 cervical nerves are going to innervate neck muscles, diaphragm, deltoid, the wrist, triceps, and fingers. So you're going to have function going, sensory function coming from these regions, and then also motor function going to those areas. Then in your thoracic region, this is where you're going to go directly into those intercostals and then one of the hands, or part of your hands. And then the lumbar region, this is going to go to the lower body, so the hips, the legs, the feet. And then the sacral region, which is absolutely critical, it's going to go to reproductive organs, and then also your ability to urinate and then to defecate. When we look at how the actual nerves are organized, they put them in groupings called a plexus. So a plexus is just an organized cluster of nerves. And we have 4 of these: the cervical plexus, the brachial plexus, lumbar plexus, and the sacral. And we do not have a thoracic plexus because they're not bunched together in terms of how they function. So the cervical plexus, what you need to remember is the range of nerves that are included in the cervical plexus, which is C1 to C5. And what they do is they innervate the neck and the posterior portion of the head. So what you're looking at here is C1, C2, C3, C4, C5. And what you see is that this is the actual root of the spinal nerve, and then they're going to branch, all right? And where they're branching off into is going to be into these portions of trunks and roots, and then they just get smaller and smaller. So this cluster here is going to innervate just this upper portion here of the neck itself, and then a very critical one that you have to remember is right here at C3. You see how it comes down and it connects to this guy right here that's called the phrenic nerve. So you've got, boom, right there. Phrenic nerve directly innervates your diaphragm. So if somebody has an upper cervical cord injury, always keep in the back of your mind that this person's respiratory rate could directly be affected. And their ability to breathe deeply, which is what your diaphragm allows you to do, to take a breath in, is probably going to be affected. So this is why it's really important if somebody is having difficulty breathing and you suspect they have some kind of cervical injury, that you take precautions, right? You do that spinal immobilization. And this is why it's also so critical when you're doing your trauma assessment that you're looking for those things, right? And checking to see if that is happening. So the actual phrenic nerve, its first point of origin is going to be coming from that C3. And then you'll see how there's an overlap here with C4 and then also with C5. So between C3, C4, C5, all of these are responsible with some of the function of what's going to happen in that phrenic nerve that then directly innervates the diaphragm. When you look at sensation, all right, this word here called dermatome. Dermatome is basically a map of sensation on the skin. And why this matters is, again, when you're going through and you're doing a trauma assessment, right, and you're speaking with your patient if they're conscious or if they're not, what you're doing is you're checking to see, do they feel this? And so where you see the V, this is for cranial nerve number 5, the trigeminal. Remember, this one has 3 branches. And so the anterior portion of the head is the first branch of that trigeminal nerve that is going to carry that sensory information. Below that, in the maxilla, in here, that's the second branch of the trigeminal nerve. And then along the jaw and the side of the face and the upper portion and the lateral part of the skull, this is going to be the third branch of the trigeminal nerve. Then with your cranial or your cervical nerves, now you have C2, C3, C4, and C5. So the back portion of the head, that is C2. Then you have C3, that is going to be just below where that C2 region is, underneath the chin and anterior portion of the neck. And then your C4 just below that, and then C5 right in the upper part where the trapezius is, right where those cervical thoracic vertebrae meet up. So those are your facial dermatomes. The brachial plexus, this one is going to innervate into the arm, so that is why it's called brachial. So it's going to hit directly into the shoulder, so where the deltoid is located, down into the axillary region, into the brachium, into the antebrachium of the forearm, and then directly into the hand. So the brachial plexus runs from C5 to T1. So in this case, you would have an individual, like if they had an injury at, T1, right? They're still going to be able to move their head side to side, right? They might still have the ability to lift, elevate their shoulders and that, because that's primarily governed by the accessory nerve. But they're going to have a very hard time probably being able to grip, right? flex their wrist, extend their wrist, maybe flex their elbow. So these are all things to kind of keep in mind when you're doing that trauma assessment, is where could that injury be? And this is why when we say, when you're doing your trauma, don't just say the words pulse, motor, sensory. Like, you're doing it. You are checking for any damage that could potentially be impacting that spinal cord. And so from C5 to T1, that is that overlap with the cervical plexus at C5 that then will feed in and give rise to the axillary radial median, musculocutaneous, and the ulnar nerve. When you look at the dermatomes, what we're looking at here, here's your C5 with your overlap, and then here comes C6. So this is going to be the lateral portion of the arm that goes all the way down to the thumb. Then you have C8, which is running down more the posterior side. And then with your C7, which is going down the midline of the arm. And then when you look at the actual hands, you can see that C8 governs pinky and finger, and then your index finger and middle finger are governed by C7, and then C6 is the thumb. So this is one of the easy ways that you can actually assess. If you tell them to grip, right, and they can actually grip, then you have motor, right? Can you feel it? Yes. All right, so we know that these areas up at the cord are actually intact. But then if you're running through and you're touching back here, right, they can feel that, but now they can't feel you touching here, then that's going to tell you that the injury is actually below C5. And so these are also things to keep in mind in terms of what's happening with you and any kind of cervical disc herniation that can then impact and cause impingement or herniation or problems with the actual nerves. So when you're feeling any kind of tingling that's happening here, or you have neck pain that now you feel tingling down in your fingers, right, depending on where it is, it's very possible that you could have a herniation that is now causing problems at that cranial nerve or cervical nerve level with your hand. And so these are real telltale clues of when things start happening. So you not only have the pain in the neck, but you're also going to have the problem down in the hands, and then in some cases in the wrist. And then hopefully not in the elbow, but it could be there too. So things to keep in mind, because of the nature of the job, you're much more at risk for having a herniation in that cervical region. So as you run through here are the cervical plexus and the brachial plexus, the nerves of what come off of there, and then what they actually do. Then when we look at the lumbar and the sacral region, the lumbar plexus is going to run between L1 and L4, and then the sacral plexus runs from L4 to S4. Collectively, these are called the lumbosacral plexus, and so what we're looking at now is lower body. The big nerve that comes off of this one, obviously, is the sciatic nerve, and that sciatic nerve runs all the way down to the back of the foot, and it's actually two nerves that are put together, right? It's basically the merging of the fibular nerve and the tibial nerve. So anteriorly, when you look at it, all right, so here are your lumbar vertebrae. So we're coming down between L1, L4, and then from L4 down to the sacral portion with your S4. These are all the nerves that are going to be affected. So this is the anterior portion. And so if there is some kind of herniation occurring up here where that sciatic nerve is going to be coming through, then that is why you can actually have pain laterally in the hip, sometimes anteriorly in the leg, and in some cases, if it runs all the way back, this is where that sciatic portion comes down. This is why individuals will have that pain all the way down through the back into their leg, and then in some cases going all the way down into the foot because of what the sciatic nerve is. And so when you look at the sciatic nerve and look at what it is, this is actually part of the tibial nerve, and then what you look at when you see the actual fibular nerve right here. So you see how this kind of continues, and then you've got the tibial nerve. So it's the portion, this sciatic nerve is part of, when it separates down at the back of the femur, it's the fibular and then the tibial nerve. Then in terms of your dermatomes, thoracic region, obviously those are thoracic nerves. We're not talking about those yet. The lumbar plexus, most of the anterior portion of the leg is run by the lumbar nerves. So you're going to have L1, L2, L3, L4. So if you start up at the top, more medially, right, that's going to be L1. Just below that, L2, and then down the midline portion of the thigh, that's your L3 to the inside. And then L4 runs through the knee all the way down to the big toe. And then when we start overlapping with the sacral, now we're looking at the anterior portion going down into the feet and the middle toes. And then with your sacral, now we're looking down at the lateral portion of the leg into the pinky toe. And then the posterior portion of the leg is mostly all sacral. So now what we're looking at is the butt and then down the back of the leg. And so what happens, this is why in these dermatomes, if somebody has an inflammation of that sciatic nerve, in some cases, when you touch that spot, it can be so painful. And then the other thing that's really important about these sacral nerves, as you see, is that here is the anus, obviously here is the genitalia. So if there's any portion of these sacral nerves that are actually damaged by some kind of pelvic injury, or if the injury was further up in the lumbar region, that individual is not going to be able to squeeze their butt, which you'll see when they're doing the trauma assessment at the hospital in the trauma bay. The docs always, if the patient is conscious, they will check to see, do they have that ability to do that, right? Or do they have that reflex? And also, is there urinary incontinence, right? What is happening there with that individual? Did something happen there? So those are things that are governed by these sacral nerves. And so here is the lumbar plexus, the keys to this, and then the sacral plexus, what part it innervates, and then what the person would present this if they don't have the ability to be able to actually move it. Then when I talk about the thoracic nerve, the thoracic portion is not a plexus, but they're numbered individually T1 to T12. Obviously T1 is part of the brachial plexus, but what we have here are ventral branches and dorsal branches. So these actually come off at the cord, and they go around and they wrap they actually run under the inferior inner portion of the rib. So this is why you never want to stick anything from the bottom up into the rib or the intercostal space, because you could actually damage these nerves. And so what's important about this is that these are the nerves that are going to supply the intercostal muscles, and then also muscles of the anterior and lateral trunk. That's your ventral branches. And then the dorsal branches are going to supply the posterior, on the back, skin and muscles of the posterior trunk. So when you look at this, here's your spinal cord. Here are the spinal nerves coming off. These are the dorsal branches going to the posterior part of the trunk. And then these are the anterior branches. And this is what I mean by they run. Your rib is going to be running through right about here. And then you have the intercostal muscle, which is going to run in between the ribs, but then inferior and inside that medial aspect of the rib, this is where that ventral branch of the intercostal nerve runs. And then you also have the intercostal arteries, so you're going to have arteries and veins running through there. So that's why we call that the neurovascular bundle. And that is why, for example, when you learn how to do a needle decompression, you will learn that you go through the intercostal space, but it's a top to bottom. So you start at the top, superior to inferior, when you're doing the decompression with the needle and then threading in the catheter. You never go from the bottom up. And then when you look collectively at the spinal dermatomes, obviously you can also see that if there's a thoracic injury, so anywhere at the cord and the thoracic region, that is where somebody would lose sensation, right? So this is why on the anterior portion of the body and posterior, right, you're going to have the thoracic nerves that you can actually ask the person, do you feel something? But why, again, T1 is part of that brachial plexus. That is why you feel sensation all along the posterior portion here of the arm, and then also slightly on the anterior portion and medial portion of the arm from that T1. So it'll be helpful for you to learn these dermatomes. It will help you very much with trauma assessment and also give you an indication, again, not only if the person's having pain, but obviously if they don't have sensation or pulse motor sensory when you're checking those trauma patients. Then flaccid paralysis, what that refers to is when you have damage to the ventral root. And so what is happening here is that there's actual damage that is taking place at the portion of the neuron that is going to go and innervate the skeletal muscle. So when that happens, is that muscle is not going to be getting the signal, which means there's no voluntary movement. They can't move that muscle, which means that joint obviously is not going to be able to function. And then eventually what happens is the muscle atrophies because they are no longer being stimulated. So that's flaccid paralysis. When you have what's called spastic paralysis, what this means is that the muscles themselves are still functional because they function through a reflex, right? A spasm. But what occurs is that the actual signal that is going from the connection from the brain to that portion is affected. And so now we have an injury that is impacting the ability for the signal to go to that muscle itself, but we have these reflex arcs that can take place that are just passing through that portion of the cord. So just above that, you have spinal reflex arcs that works. This is why sometimes you'll see individuals that have bungee cords on their legs because they have this type of spastic paralysis that just causes their legs to spasm, and in some cases they could just kick out. And so that is what ends up happening with spastic paralysis. And then individuals that are quadriplegic, obviously the injury is going to be at T1 or above, and that would be quad, so 4 of the limbs are going to be impacted. And then paraplegic is going to be below, so anywhere from T1 and down, or T2 and further down. So paraplegic means you still have function of the arms, but anything below that in the legs are no longer working. So the way that the reflexes work is that you're going to have somatic and autonomic. And so the somatic ones are going to be the ones that stimulate the skeletal muscle, and the autonomic are going to deal with smooth muscles, the heart, and the glands. And so what you're looking at here is you're going to have basically a 2-neuron reflex arc, with the little interneuron is going to be in the middle. So what you're looking at here, this is probably a real simple one, right? So signal comes in, gets processed, and it goes right back out. So this would be an easy example of a somatic reflex. Here is another one where you're going to have the interneuron, right? So the signal comes in, gets mediated, and then goes back out to the effector organ. And then when you have what's called a somatic multisynaptic reflex, then what we have here is the signal is basically you stepped on that thing that was bad. Now you're going to come in, signal comes in through the sensory, and this one's really complicated because now you have what we call multisynaptic reflex arcs. And so in this case, what is going on is that not only is it happening on the same side of the body, but it crosses over to the other side to help you stay standing up so that you don't fall over. And so in this case, you have interneurons that are going to be mediating the same side of the body and then also the opposite. So this flexor withdrawal reflex means you flex the knee, you flex the hip, you withdraw from the danger. Signal got sent that caused that withdrawal to take place. And at the same time, there was a crossover to the opposite side so that signals would get sent to the muscles that are going to actually stabilize and stay extended, keep the leg extended so that you don't end up hurting yourself. So this is probably the most complicated of the type of reflexes that we have. Then when you look at the autonomic reflex, now we're looking at how is it that, say, blood pressure is regulated or digestive system. So now what we have here, this is a response. Basically what you're looking at is what happens with the sympathetic response. So autonomic, here is the lateral horn of the spinal cord where the sympathetic, what we call the preganglionic neuron, comes out. And it's going to go through and it's going to meet up in a ganglion. Remember, a ganglion is a swelling in a nerve that is going to connect to the second neuron that will then go to the organ. So in this case, what we're looking at here, this is, for example, your small intestine. So when your sympathetic system fires, you want to inhibit what is taking place in the digestive system. So this would be an example of how that's able to do that. What we're going to talk about next here with the autonomic nervous system, it's going to tie in directly to how it is that these somatic reflexes or autonomic reflexes actually work. And so what we're going to focus on now is going to be down here on the autonomic division of the nervous system that ties into your parasympathetic and your sympathetic. So we're going to be right here. And then the main thing that you want to remember about this is that with your sympathetic, you're looking at your fight, flight, or freeze. And then anything that's a shock, anything that's stress, anything that's going to be perceived as being threatening to your resting state. All right? And then that is also when we kick on the adrenal glands that are going to be responsible for secreting epinephrine and cortisol. The parasympathetic system, again, that is your resting homeostatic state when you're in complete balance and everything's at rest and relaxed. So when you look at how it is that our neurons are organized to be able to manage these two systems, you'll notice here on the brain that the parasympathetic side, which is signified in this kind of purplish color, the origin of the first neuron that then connects to a second neuron, because this system is a 2-neuron sequence mechanism, the parasympathetic, the origin of what we call the preganglionic neurons, the first neuron in the series, starts up in the brainstem, and it also has branches in the spinal cord, or in the sacral spinal cord. But there is nothing in between. What's in between is the sympathetic side. So the sympathetic portion goes from T1 down to about L2. And so what you're looking at here is your sympathetic neurons are originating from a completely different place in the body in the central nervous system. So these guys, the sympathetic, go from T1 down to about L2, and then your parasympathetics, the origin is from the brainstem and the sacral portion of the spinal cord. But you will see that they're both going to the actual organs that they are both going to end up affecting, which all have autonomic functions. With our sympathetic system, one thing that I wanted to point out when I brought in this image is so that you could see what we mean by a ganglion within the body. So in your parasympathetic system, this originates from the brainstem and then will go to the organ, and the actual connection where it meets up with that second little neuron, postganglionic neuron, is actually at the organ itself. With the sympathetic branch, what ends up happening is that what you see here with these little circles, they're just signifying what a ganglion would be. And so what occurs is that we actually have, this right here is our vertebrae, what is running here through the middle. And then coming off the vertebrae, as you know, on the posterior side, you have spinal nerves. Well, on the anterior side of the vertebrae, just running parallel to where those spinal nerves are going to exit, we have this thing that's called the sympathetic chain, and that's what you're seeing here. And all these little areas here where it looks like a little swelling, okay, so it's a little thicker here, a little thicker here, a little thicker here. All these little bumps are ganglion, are the little ganglia that actually exit and that are the location of where that preganglionic neuron that comes off from the spinal cord is going to exit and then make up synapse, basically right here, and make its connection to the postganglionic neuron that will then travel out and go to where it needs to go to the organ. So what you're seeing here is actually from a cadaver, and this right here is called the sympathetic chain. And this is where the two neurons of the sympathetic system actually meet up and make their connection. So what I'm talking about here is that your body organs, right, the ones that are going to have the autonomic function that you don't control and you don't have any voluntary activity over, they are controlled by autonomic functioning. Both organs, so like say your digestive system, is going to have connections from both sympathetic and parasympathetic. However, sympathetic fibers are only going to your blood vessel muscle, sweat glands and hair follicles in your skin, and what we call the adrenal medulla. These pieces do not have parasympathetic connection. So your sympathetic system is what's controlling this. And when we end up talking about the cardiac system and then what happens with blood vessels and blood pressure, this will come more into play, and hopefully it'll make a little bit more sense. But these connections for blood vessels, sweat glands, and the adrenal medulla of your adrenal gland, where epinephrine is secreted, only are mediated by the sympathetic system. So when we look at what we call the two-neuron system, what happens is that both sympathetic and parasympathetic have a preganglionic, so what you see here in the green, and a postganglionic neuron. So what has to happen is this preganglionic neuron that you're seeing here will secrete its neurotransmitter onto the postganglionic neuron, so what you're seeing here, and then this neurotransmitter will land on these receptors that you see on the postganglionic neuron and stimulate the postganglionic neuron. And then at the end of the postganglionic neuron, it will then secrete a neurotransmitter that then ends up affecting the target cells of that organ. So what I'm going to do is I'm going to go through and talk about what these neurotransmitters are, what the receptors are, because they're very specific, and then what the target organs are. So we're going to start with sympathetic. So your sympathetic system, remember, is coming off T1 down to about L2. So the preganglionic neuron is going to originate in the lateral horn of the spinal cord. And then what it's going to do is go out and find that sympathetic chain, which is only for the sympathetic preganglionic neurons to go and connect to the postganglionic neuron that then travels to the organ that it's going to innervate. So in this case here, for example, at T1, we have the preganglionic neuron that will travel out, and then it'll make a connection with this postganglionic neuron that then goes up and innervates the eye. So in this case, this postganglionic neuron will cause your eye, your pupil, to actually inhibit the parasympathetic response that maintains it in a normal resting state. And when norepinephrine is secreted here, what causes is it dilates. So it actually relaxes the muscles that are in the pupil to allow it to open up. Next spot here, boom, it makes its synapse. It's going to go to the connection to the postganglionic neuron that innervates the salivary glands. Because saliva secretion is a parasympathetic function, when your sympathetic system kicks on and secretes norepinephrine, what it's going to do is it's going to inhibit salivary gland secretion. Then with our heart, when you need to speed up, increase heart rate, increase contractility, all of that stuff, here comes our preganglionic neuron, connects to this plexus, and it goes to the heart and then causes the heart rate to increase. Then when it connects here to the lungs, what it's going to do, it's going to be able to increase the ability for us to bring in more air because we have more opening of our bronchi and our bronchioles, and it also allows for the lungs to be able to expand more. Then down here with our digestive organs, what ends up happening is the nerves, the preganglionic neuron actually leaves, and its connection, it finds a ganglion that is actually outside of that sympathetic chain. These are called the celiac, mesenteric and superior mesenteric and inferior mesenteric ganglia. Now, I'm not going to test you on these specific ganglia out here, but what these are doing is that this is where the connection is for that postganglionic neuron that is then going to go and connect to the digestive system, the kidneys, and the reproductive organs. And when your sympathetic system kicks on, basically what it's doing, it's going to inhibit function in digestion and urinary formation. What's also going to happen is that it's your sympathetic system that is responsible for, within the genitalia itself and the reproductive system, for the climax and the sympathetic response for ejaculation in the male and orgasm in the female. And so when we get to the reproductive, we'll talk more about that. But for now, what I want you to understand is what the sympathetic system does is that it's going to kick on sympathetic functions that are all about fight, flight, right? So they're responding to some kind of stress. And so what happens is, here's your lateral horn, all right? So remember, you have your preganglionic neuron comes out, and it's going to synapse within a ganglion. So the synapse can actually be within the sympathetic chain, either a little further up from the parallel position of where it originates in the spinal cord, or it's going to be exactly parallel to where that is, where it synapses to another postganglionic neuron. It can also come out and then leave, so just pass through the sympathetic chain and then find the ganglion that is just outside, synapse with its postganglionic neuron, and then travel to its target organ. So with your sympathetic neurons and receptors, the preganglionic neuron, I made it green here because it's green on the diagram, this is going to be a neuron that we call a cholinergic fiber, and the reason why is because it secretes acetylcholine. So what that means is that this preganglionic neuron is going to secrete acetylcholine, and the receptors that it's going to secrete it onto are cholinergic receptors. The postganglionic neuron, in this case, can secrete two things, either acetylcholine or norepinephrine. This is a unique thing about the sympathetic system. So when it secretes acetylcholine, this postganglionic neuron is cholinergic. When it secretes norepinephrine, now the postganglionic neuron is considered to be what we call adrenergic. So if it secretes acetylcholine, which is here, that is a cholinergic neuron because it secretes acetylcholine. And with the sympathetic system, what it's doing is acetylcholine actually stimulates the receptors here that are called muscarinic receptors on the sweat glands. If this was an adrenergic fiber and it secretes norepinephrine, then the receptors are called adrenergic receptors, and they are receiving the norepinephrine. So we'll get into that next. The parasympathetic branch, remember, is what we call craniosacral. So what that means again is that now we're going to be secreting from the cranial portion of the brainstem. These are where the preganglionic fibers are going to come, and they're going to meet up with a ganglion that is very closely located to where the actual organ is that is the parasympathetic branch that is going to be innervating it. So you'll see that here with these three, with the 4 cranial nerves that actually do that. Cranial nerve number 3, oculomotor. Cranial nerve number 7, facial. Cranial nerve number 9, glossopharyngeal. And then cranial nerve number 10, which is the vagus nerve. Then down below, the sacral preganglionic fibers will come out and they actually connect to what you're going to have with the bladder and the genitalia. The parasympathetic neurons, these are going to be cranial nerves 3, 7, 9, and 10, like I just showed you in the image, and then the sacral nerves that go through S2 and S4. And these are cranial nerves that I want you to remember that are part of the parasympathetic function. So if I ask you on the test, which of the following parasympathetic or cranial nerves are part of the parasympathetic function, remember that it's number 3, oculomotor, 7, facial, 9, glossopharyngeal, and 10, the vagus. So the way the parasympathetic neurons function is you have your preganglionic neuron secretes acetylcholine, so that makes it a cholinergic fiber. The postganglionic neuron secretes acetylcholine, that makes it a cholinergic fiber. So both of these secrete acetylcholine. So what you're seeing here is the preganglionic neuron secretes acetylcholine, and on here, these are nicotinic receptors. That's very important to remember. All right? At the postganglionic neuron, the receptors for acetylcholine are called nicotinic receptors. The postganglionic neuron, also a cholinergic fiber because it secretes acetylcholine, then is going to target the organ to cause a parasympathetic response, but the receptors here are called muscarinic receptors. So, the two types of receptors that we have for your parasympathetic system that are cholinergic receptors are going to be nicotinic, which are always excitatory, and muscarinic, which can be inhibitory or stimulatory, depending on what organ they have. What also I want you to note is that acetylcholine esterase, this is the enzyme that cleans up acetylcholine from the synapse, where the neuron that secretes the acetylcholine meets up with where that target organ is going to be. So looking back to the pictures, what you're looking at here is preganglionic neuron secretes acetylcholine. Here we have nicotinic receptors, which means that this preganglionic neuron on the nicotinic receptor is going to cause this neuron to be stimulated. This was sympathetic. Parasympathetic, preganglionic neuron secretes acetylcholine, nicotinic receptors stimulatory. So in the middle, in both your sympathetic and your parasympathetic function, where the two meet up with the postganglionic neuron, acetylcholine is always secreted, and the receptors here in the middle are always nicotinic receptors. So what that means is you will always have stimulation of the postganglionic neuron in the sympathetic junction right here and in the parasympathetic, because these receptors are nicotinic. Why this matters is you would not want this preganglionic neuron in the sympathetic or the parasympathetic to secrete acetylcholine and these receptors here not respond. So that is why these nicotinic receptors are always stimulatory and they will always respond. When your sympathetic system, when the fiber that comes from the lumbar region and goes to the adrenal medulla, which is where epinephrine is secreted from, that adrenal medulla actually has nicotinic receptors on it so that it will always secrete epinephrine. All right? The adrenal medulla is kind of like this big ganglion. When you look at what's a muscarinic receptor, here's how it works. With your sympathetic system, your preganglionic neuron secretes acetylcholine. Here are the nicotinic receptors, always stimulatory, stimulates the postganglionic neuron, and when you have this postganglionic neuron in your sympathetic system that secretes acetylcholine, that means that it's targeting a sweat gland. Just think about it. When you go into a sympathetic state, you will trigger sweating, right? Because it's a stress response or a response to help cool you off. So the target cell is going to have cholinergic receptors. The type of cholinergic receptor on the target cell is a muscarinic receptor. In this case, the acetylcholine secreted here on that target cell, when this fiber is cholinergic, is going to be a muscarinic receptor, which means in this case it's going to be stimulatory. So it stimulates the glands to secrete sweat. In the parasympathetic system, now what we have with our target cell is a little different. So when we are in a rest and digest state, the preganglionic neuron secretes acetylcholine on the nicotinic receptors, stimulates the postganglionic neuron, it secretes acetylcholine, and that acetylcholine targets the muscarinic receptors. If we are in a parasympathetic organ that is going to function and be stimulated under rest and digest, which means your digestive system, your reproductive system, and the formation of urine, these muscarinic receptors will function as stimulatory or excitatory. But if it's the parasympathetic neurons that will then connect to the postganglionic neuron that is on your heart and on your lungs, this one is now going to secrete acetylcholine on the heart, and in this case the muscarinic receptors of the heart are actually going to be inhibited. So now what that does is that it actually causes your heart rate to slow down, and how it does that is that it causes more potassium to leave your heart cells and basically hyperpolarize your heart, so it decreases the number of cells that are beating and it slows down your heart. When we get to the cardiac system, that's where I'm going to talk a little bit more about this. So when you get to summarizing this, the cholinergic muscarinic receptors, remember they are located on the target organs, and if they are excitatory, that means if you have the sweat glands are going to contain excitatory muscarinic receptors. So they're going to cause you to sweat. Digestive organs have excitatory muscarinic receptors, so it's going to have digestion that is going to occur. Reproductive organs, urinary organs, same thing. If the cholinergic muscarinic receptors are inhibitory, then that means that they are going to be on the heart and on the lungs, right? They're going to decrease the ability to ventilate and decrease respiratory rate. So when we look at what we have on sympathetic, when your sympathetic system is now secreting adrenaline, which is your norepinephrine, now the type of receptors that it targets are going to be what are called adrenergic. So now we have adrenergic receptors. We have two subtypes, alpha receptors and beta receptors. This is what they look like. So norepinephrine gets secreted from adrenergic fibers. The adrenaline is going to target the organ, and it's going to target the organ, and the receptors that are going to receive the norepinephrine are either called alphas or betas. Monoamine oxidase is the enzyme that cleans up norepinephrine. So when we move to here, the alpha receptors that we have are alpha 1s and alpha 2s. Alpha 1s are the ones that we really want to focus on. That is why I kind of made this a little bit lighter with the alpha 2s. So when you have a sympathetic response, you have your preganglionic neuron secretes acetylcholine in the middle, like always, on these nicotinic receptors that are on the postganglionic neuron. Now, because norepinephrine is being secreted out here at the end of that postganglionic neuron, this postganglionic neuron is an adrenergic fiber. So what that means is that here, which is the muscle of the arteries and the veins, are alpha 1 receptors. And so what's going to happen here is that when norepinephrine lands on these receptors of our blood vessels, it causes them vasoconstriction. So they constrict and increase pressure. So when your body needs more systemic pressure, what is going to happen is your sympathetic system kicks on, that postganglionic adrenergic fiber secretes norepinephrine. Those vessels have the receptors, alpha 1 receptors, and they're going to cause constriction, which means that it's going to increase pressure in the vessel. The alpha 2 receptors, these are located on the gut and within the kidneys. So what's going to happen here with your alpha 2s, what they're doing is you're actually inhibiting parasympathetic function. But why I want to primarily focus on alpha 1s is that this is how your diastolic blood pressure is primarily regulated. And so the vessels, your veins and your arteries, have these alpha 1 receptors, so that pressure will increase when you're in a sympathetic state. With the beta receptors, we have two subtypes, beta 1 and beta 2. So if we have our preganglionic neuron, targets, meets up with the postganglionic neuron, we secrete acetylcholine here, nicotinic receptors, always stimulatory, and then the adrenergic fiber is going to secrete norepinephrine. On the heart, what is going to happen is that it's going to target beta-1 receptors. What that does is it increases heart rate. It speeds up the conduction system. It increases contractility. It makes everything ramp up so you can meet the needs of blood flow and distribution from a sympathetic response. The beta-2 receptors are located on your lungs. So, preganglionic neuron secretes acetylcholine, targets the nicotinic receptors, always stimulatory, causes the adrenergic fiber to secrete the norepinephrine on the lungs. The lungs have the beta-2 receptors that causes the bronchial dilation, opens them up, and it allows for more air to be able to move in and out. And so you're able to get the necessary oxygen in and then able to get the carbon dioxide waste out. And so that is how it is that your sympathetic system runs through with these beta adrenergic receptors. What you're looking at here is basically the connection of what happens with the heart. And so how you have your parasympathetic and your sympathetic system that are able to control this has to do with they have dual innervation, which means you have sympathetic and parasympathetic function. This right here is the origin of where that vagus nerve is going to be. So this is your preganglionic neuron from the vagus nerve coming down, innervates the heart, connects to its postganglionic neuron that connects to the two little areas that basically are going to regulate heart rate. This is your SA node, this is your AV node. And so when acetylcholine is secreted on these two little buttons, basically what you're doing is you're slowing down heart rate. Now when we go into a sympathetic response, we have the function that comes right off here from the lateral horn of the spinal cord at T1. We have the neuron, the preganglionic neuron meets up with the postganglionic neuron here, and then that postganglionic neuron connects to the 4 spots on the heart instead of just 2, so we can very quickly speed up heart rate at the SA node, speed up the signal that is passing through the AV node into the ventricles, and also we have direct connection from sympathetic branches that go to the ventricles so that everything in the heart can very quickly speed up and respond to that sympathetic need that we're having. When you take an anticholinergic medication, what is going to happen is that these are going to block the effect of acetylcholine. So essentially what you're doing is you're blocking parasympathetic response on the heart. And so what this does is that it's going to help the heart speed up. So this would be a case when somebody is having a very, very slow heart rate. Atropine is one of the medications that you provide in order to help the heart speed up. What it's also going to do is that it's going to prevent salivation, and it also dries up respiratory system secretions. So what you're doing here is you're basically giving them a drug that acts like a sympathetic response and thereby blocks the parasympathetic. When you give a medication that is more sympathomimetic, what that means is now you have a drug that is going to mimic sympathetic, for example, norepinephrine and epinephrine, right? Now you have a medication that can target alpha receptors, so increase blood pressure, and also target and stimulate beta receptors, which means we can speed up heart rate, we can speed up the ability to ventilate and move air through, right? And we are inhibiting parasympathetic responses, so we're inhibiting salivation, we are inhibiting digestive function, urine function. So when you look at this, atropine, because it's an anticholinergic, this is also the drug that we give when somebody has organophosphate poisoning or has been exposed to a nerve agent that basically what it does is it keeps acetylcholine around. And so because acetylcholine is what stimulates digestive function, it's what helps you poop, it's what helps you pee, it's what causes your eyes to tear, it's what causes salivation. When you have some kind of organophosphate poisoning or nerve agents that keep acetylcholine around, they're not allowing it to be cleaned up because they're blocking that enzyme acetylcholine esterase. That is why you end up with the person basically leaking fluid, right? Atropine is the rescue medication for that, and you have to give it in pretty high amounts, and then what you're doing is you're trying to reverse the effect of the acetylcholine. So that is, and you'll learn more about these as you guys start going through and learning the drugs. So to summarize it in terms of how overall the whole autonomic nervous system works. Remember that you have two neurons that are involved. You have a preganglionic neuron and a postganglionic neuron. With your sympathetic system, the preganglionic neuron is going to secrete acetylcholine always. Always secretes acetylcholine here. The receptors on the postganglionic neuron here are going to be nicotinic receptors that are always stimulatory. Here on the adrenal medulla, which is the core of the adrenal gland, we have the nicotinic receptors that are going to respond and will stimulate the secretion here of epinephrine. Then, if this postganglionic neuron is going to the sweat glands, it's a cholinergic fiber and it's going to secrete acetylcholine and stimulate sweat gland production. If the postganglionic neuron is adrenergic, it's going to secrete norepinephrine and it's going to target on the target organ alpha adrenergic receptors, alpha ones, increase blood pressure, or it'll target beta receptors, beta 1 in your heart, or in the lungs, the beta 2s. And then it's going to inhibit, when norepinephrine gets secreted, it will inhibit digestive function and also, obviously, urinary function. With your parasympathetic system, the preganglionic neuron is cholinergic, secretes acetylcholine, meets up at the ganglion. The postganglionic neuron here will have nicotinic receptors. The nicotinic receptors will cause stimulation of the postganglionic neuron, which is a cholinergic fiber, and it will secrete acetylcholine on muscarinic receptors, which can be either stimulatory or inhibitory. And so what we have is a system that is able to control similar organs, right, but have opposite effects on them. Here with your somatic motor, what we have here, this is your voluntary control. And so with your somatic motor, this neuron is one neuron that leaves the spinal cord, happens to at the end also secrete acetylcholine, and the receptors that we have on skeletal muscle are nicotinic receptors, which means that every time acetylcholine lands on skeletal muscle, it's going to stimulate muscle contraction, which is what you want, right? Because you want to move a skeletal muscle. So that is what's occurring with these systems. So with these next tables, what you're seeing is that they actually have the organ system, what the parasympathetic system does, and what the sympathetic system does. So you're looking at the effects of these, all right? And so that's the end of the nervous system. Let me know if you have any questions. I'm happy to answer these, and reach out if you need help.